All right, team, here we are again. This is the next video for this unit, which covers material for the second test. And just to show you where we are, or where we're about to be, we were down here in energy, and we did energy, photosynthesis, cellular respiration, PowerPoint. You can always go back and look at that, or if you just watch the video, you'll see it. Now we're going to jump up here into module 2, or module 3 rather, the organization of matter into cells. And we are going to be in this PowerPoint that says the cell cycle, mitosis, meiosis, PowerPoint. Okay, so this lecture will cover the cell cycle, mitosis, and meiosis. We'll see how far we get. Okay. So what we want to understand a little bit about now is how do cells reproduce? That's really what we mean when we talk about the cell cycle. So, you know, the thing to consider about a cycle is it goes round and round, right? So this gives you an example of a cell cycle. And if you look in here, you can see we actually use these abbreviations, G1, S, G2, and M. And so we want to go through and talk about these different parts of the cell cycle. The idea is if we start right here at G1, we have a little baby cell, let's say, a cell that was just formed. And anytime you have a cell that was just formed, you're going to be automatically in G1. The G stands for gap. This is gap 1. And you can see that it's called the first growth phase. It says growth and normal metabolic roles. And so this little baby cell starts to grow and develop its normal metabolism that you would expect. Okay, that's G1. The S phase is, the S stands for synthesis, and you can see that right here. And the idea is if we're going to make more cells, then what we need to be able to do first is replicate the DNA molecule. And so what's being synthesized is in fact the DNA. And we call this DNA replication. So the S phase or synthesis phase is where the DNA is replicated. So it's worth it to take a second to understand what that means by replication. You know, what do we mean? Well, what we mean is that we start with one copy of the DNA and we end up with 2 identical copies or replicas. Okay, so we're going to have going from one set of chromosomes or one complement of DNA in the nucleus to 2 complements that are identical in every way, barring any sort of mutation that could potentially occur in the replication process. We're not going to do the replication process in detail in this unit. We actually do that when we do protein synthesis. And so we're going to learn more about it. But for now, just suffice it to say, you know, you're making 2 copies of the DNA from one copy. Okay. Then we have the G2, gap 2, which is the second growth phase. And this is growth and particularly preparation for this process that we call mitosis. Okay, so what is mitosis? Well, we're going into it here. The M stands for mitosis. And you can see we break it up into 4 parts: prophase, metaphase, anaphase, and telophase. I'll give you a little hint. If you just remember the PMAT is it's the little rug beside your toilet. It's the PMAT. Everyone has one in their bathroom, right? You're not supposed to pee on it, but you might stand on it while you're peeing. Prophase, metaphase, anaphase, telophase. This gives you an idea of, it reminds you of the order of these 4 phases. So in just a bit, we're going to go through and we're going to walk through mitosis and learn about what's actually happening in a cell in these 4 different phases. Okay, so a few things. G1, S, and G2 have been called interphase. Okay, so interphase refers to these first 3 phases. So the entire cell cycle, you could say, consists of interphase plus mitosis. They used to refer to interphase as the resting state of the cell, which is really a poor thing to call it because the cell is not resting at all, right? It's growing, it's replicating its DNA, it's growing some more and preparing for mitosis. So we call it interphase. Okay, so that completes the cell cycle. Now we need to understand a little more about mitosis before we go on and actually go through the phases. So let's talk a little bit more about mitosis. Let me see what this next slide is. Okay, so there's never really a good spot to do this, and so we might as well just do it now. Okay, it is really important for you to understand the difference between haploid and diploid, and it's really very simple. It doesn't have to be confusing to understand the difference. We use the word haploid. We also abbreviate it as 1N. Or the word diploid, di means 2, or 2N, to represent the condition of of the chromosomes in the cell. And so now we want to think about what is a chromosome, okay? And so there's something to think about. There's 2 forms of DNA in the cell. And one form is called chromatin with an N. And the other form is called a chromosome. Okay. We're going to see there's lots of C words in this unit. I mean, when we do mitosis, there's all these C words: centromere, centriole, chromosome, chromatin, chromatid. And it's a challenge to keep all the C words and remember what they all mean. But for now, what I'm telling you is there's 2 forms of DNA, and chromatin is the thread-like form of DNA. And a chromosome is the rod-like form. And so it's the same molecule and it contains the same information. It's just a different form of it. The best way I can describe it is if I had a big long piece of thread and I had someone else in the room and they held one end of the thread and I held the other, and we all of a sudden start twisting the thread. The thread represents chromatin. It represents the thread-like form of DNA. If we start twisting that thread in opposite directions, like this, it's going to get shorter because it's going to start wrapping around itself. And that's literally what happens. We say that the chromatin condenses to form a chromosome. And so then what we end up with is this, from this sort of thread-like form of DNA that we call chromatin, to this very much rod-shaped structure, okay? And so, you know, we often represent a chromosome like this little rod. And then when the chromosome is replicated, we often represent it as an X, like this, okay? Now, when you've seen pictures of chromosomes, like the human genotype or the human karyotype, where they show all the different chromosomes of humans, they're showing it in the rod-like form. You can't really see chromatin in the nucleus. It's very diffuse, but you can certainly see the chromosomes. Heck, you can even count them, right? And so humans have 46 total chromosomes, okay? So it's important to understand that there are these 2 different forms. And so when we're using the term chromosomes up here, you know what we're talking about. Okay. So there's that. Now here's the thing. A haploid cell has one set of chromosomes and a diploid cell has two sets of chromosomes. All the cells in your body as animals are diploid, except the only haploid cells in your body are the sperm and the egg. And of course, if you're male, you have sperm, and if you're female, you have egg. All other cells are in fact diploid. So if you look at a skin cell or a liver cell or a muscle cell, they're all going to have two sets of chromosomes. Where did you get the two sets from? Well, you got one from your mother and one from your father, right? Your father's sperm had a set of chromosomes. Your mother's egg had a set of chromosomes, and they came together to make the first cell of what became you, which is a diploid cell. Okay, so we'll talk a lot more about haploid and diploid, but it's really important that you understand the difference between the two. Okay, and so we're gonna draw some cells and look at chromosomes and try to understand what we're talking about here. Okay, let's take a second to do that. So what if I have a cell? I'm going to draw 4 cells. 1, 2, 3, 4. And we talked about replication of the DNA or the chromosomes, right? In the S phase, the synthesis phase, we said the chromosomes were replicated. Well, we can draw an unreplicated chromosome, sort of like this rod-shaped structure, okay? And we can draw a replicated chromosome like this X, like this. There's all this terminology associated with these chromosomes. We're going to say unreplicated here and replicated. So during the S phase or synthesis phase, this replication occurs. So if we look at the replicated one, let me just draw it like an X. It has all these parts to it. Here's where all these words come in. There's one replicated chromosome here. I'm going to get rid of these so it's not so confusing. Okay, we'll have a little more room. So here's what we have. They're attached to each other. And they're attached by this point somewhere in the center, and it's called the centromere. Okay, so what you're looking at here is a replicated chromosome. The replicated chromosome consists of 2 identical sides. And the 2 identical sides also have a name. And the name of the 2 identical sides, there's one side, and there's the other. And they're called chromatids. Okay, and so there's 2 of those. The chromatids are identical in every way in this case. Okay, they all, this side and this side is the 2 copies of the DNA. Okay, so what you're looking at here is a replicated chromosome consisting of 2 chromatids that are attached at the centromere. Okay, so there's a lot of terminology. Now, a replicated chromosome is not the same as 2 sets of chromosomes, and this is the point that I'm trying to make by drawing these cells. So let's use some different colors. Let's use size, though, to represent homology. When we have a diploid cell and there's 2 sets of chromosomes, we say that each chromosome is homologous or has a homologous partner. So there's a lot of terminology associated with this, as you can see. So this is a replicated chromosome, but I could draw this right here. There's one replicated chromosome. Oops. And here's another And in a diploid cell, every chromosome has a partner. Doesn't matter if it's replicated or not. I could draw the same thing unreplicated like this. And this will start to make more sense as we keep going, okay? So check it out. If we use size to indicate homology or homologous partners, then these two are partners. Unreplicated chromosomes are partners. These two replicated chromosomes are partners. So what if I draw some cells now? Let's say these are cells. And what if I draw this? Remember, we're letting size represent homology. Okay. And what if I drew it like this? And I can ask you 2 questions about each of these cells. Let me draw them all and then we'll talk about it. Okay. Let me just keep going with this and you'll see what we're going to do. And you're going to understand everything there is to know about this. Okay, we have 4 cells here that I've drawn, and I'm using the terminology, to represent the terminology we've discussed in these drawings. So look at the first cell and ask yourself, is it replicated or unreplicated? Is each chromosome replicated or unreplicated? Does each chromosome have 2 chromatids? It does not in this drawing, right? So this is unreplicated. But is it haploid or diploid? Does each chromosome have a partner? If we use size to represent homology, each chromosome has a partner. This one has a partner, and this one has a partner. So it is, in fact, diploid, or 2N. Okay. What about this one? Is it replicated or unreplicated? Again, it's unreplicated, right? Is it haploid or diploid? Does this chromosome have a partner? It does not. Does this chromosome have a partner? It does not. We're using size to show the partnership. So this is haploid, or 1N. What about this one? Is it replicated or unreplicated? It's clearly replicated. Each chromosome has 2 identical sides, right? So it's replicated. Is it haploid or diploid? Are these partners? They are not partners. They're different sizes. So it is also haploid. And then lastly, we have this last drawing. Are they replicated or unreplicated? They're replicated, right? Are they haploid or diploid? Well, each chromosome has a partner, so they are 2N. Okay? So take a second to just take all this in, rewind and watch this part of the video again if it doesn't make sense. Haploid and diploid is not the same thing as replicated and unreplicated. Replicated refers to a single chromosome having 2 identical sides. Diploid refers to each chromosome having a partner. And you might be wondering, what makes them partners? What makes a homologous pair of chromosomes? What makes them partners is that they have the same genes, okay? So let's write that down. That's important. Okay. Why are they partners? Is the question. And the answer is, I'm going to say homologous chromosomes have the same genes. What in the world is a gene? A gene is basically a specific location on a chromosome that deals with a trait in the organism. And if I ask a classroom of students, can you give me an example of a gene? I always get the same answer. I get eye color, hair color, maybe skin tone. These are things that we automatically associate as being genes, and they are in fact genes. They are very complicated genes. because it's not just one pair of chromosomes controlling the trait in those examples, but those are good examples. So what I'm saying is, what makes this a homologous pair, so I'll say homologous pair of chromosomes, they have the same genes. And so if this one has the gene for eye color and hair color, then so does this one. And all the genes line up. They're the same genes in the same order. Now, a couple of things to think about. Is the expression of the gene the same? Not necessarily. Where did you get these from? You got one from your daddy and you got one from your mama, right? So the expression doesn't necessarily have to be the same. In other words, you know, maybe your daddy has brown eyes and your mama has blue eyes. But the fact is the gene is still the same. The gene is still eye color. Okay? So this is what makes them a pair of chromosomes. And in a diploid cell, it makes sense that you have pairs because, like I said, the only haploid cells in living things are, the only haploid things in animals, including us, are sperm and eggs. All the other cells are diploid. Okay? So that's a lot of stuff to understand, I realize, but it's very important stuff. And so at this point, I feel like we're probably good to talk about mitosis and what mitosis is and what it means. Okay. Okay. So let us go to the next slide and let us talk about mitosis. So what is mitosis? What does it do? What's the point of it? And I'm just going to tell you that mitosis is a form of asexual reproduction. What does that mean? Well, it's hard to understand what asexual reproduction is unless you know something about sexual reproduction. The fact of the matter is, what mitosis does is it takes one cell and it makes two cells. It doesn't change the ploidy. It doesn't, in other words, a haploid cell makes two haploid cells, or a diploid cell makes two diploid cells. Both haploid and diploid cells can do mitosis in theory. In our bodies, the cells that are doing mitosis are diploid because all of our cells are diploid except for sperm and eggs. And in our bodies, sperm and eggs don't do mitosis. Okay. What's the point of it? It's just to make more cells. You know, we all started out as one cell, right? And from that one cell, we have, depending on who you ask, 40, 60 billion cells in us. So here's an example. You skin your knee in a skateboarding accident and the cells on the edge divide to fill in the scab. That would be a really good example of mitosis. So it's cell division. Nuclear and cell division is really what it is. And there are many examples of it. You know, due to injury, you might need more cells, but just in general, cells are constantly being reproduced because they wear out. So like the lining of your stomach, for example, is, or the lining of your intestines, those cells are constantly wearing out and sloughing off and new ones are being made through mitosis. Okay, so we're going to actually draw mitosis. Let's look at the next slide, but more than likely, I'm going to want to draw this out because I think it's nice. You know, we can look at this fancy drawing and there's just a lot of words and it's complicated, you know, by all the words and the definitions. But it'll be nice once we walk through for you to look at this and then it will make more sense. And we'll also watch a little short video, which is very cool. Okay. So let me pop this Windows ink up. Oh my goodness, look at that. What is this? You've already seen this video. This is where we added up all the ATPs. Got our 36 ATPs, remember, added to 38, and we had to subtract 2 as the shuttle cost, and then we had to run these NADHs and FADH2s through the electron transport chain, and that's what the 3s are about, and the 2 is about here. Okay. Good little review there. Okay. Here's what's going to happen. We're going to use size to represent the homology of the chromosomes. And I'm going to do a cell right here where 2n is equal to 4. What that tells you is that in a diploid cell of this particular organism, there's 4 chromosomes. And if this is, I'm going to draw a diploid cell doing mitosis, then it's going to look something like this. I'm going to have 2 big ones and 2 little ones. Okay, makes sense? And then we'll put those in a nucleus. And so where we are, we've already done apparently the S phase, right? Because each chromosome is replicated. So we've done G1, we've done S, we've done G2, and now we're entering into prophase, which is the first phase of mitosis, okay? So what happens in prophase? In prophase, we have a variety of things occurring. One of the things that happens in prophase is nuclear envelope disappears. Now that might seem kind of strange, but remember these chromosomes are trapped inside of the nucleus. So what I'm going to do is I'm going to show this nuclear membrane. I'm going to have to draw the chromosomes back in because they're accidentally erasing those. So one of the ways that we like to show that is by a dashed line like that. So I'm showing the nuclear envelope breaking down, okay? There's that. The other thing that happens is the centromere divides to form 2 centrioles. What in the world are centrioles? Well, I told you there was one other organelle, and we would talk about it later, and that organelle is the centromere. The centromere divides and makes 2 identical centrioles, and they migrate to the poles, what we call the poles of the cell, to either end. And we're going to see what these do in just a second. Okay. So that's pretty good for prophase. The nuclear envelope breaks down, centromere divides to form 2 centrioles. What about metaphase? Let's draw metaphase. In metaphase, we're going to say chromosomes line up on the equator of the cell. Well, if these are the poles of the cell, where's the equator? It's obviously somewhere in the middle, right? We'll draw a little dashed line across here like this. And what I'm saying is that these chromosomes just kind of randomly line up, and it doesn't really matter what order you put them in. They just kind of randomly line up along the equator. It's called the law of random assortment. They just randomly line themselves up along the equator of the cell. And then we have spindle fiber formation. Spindle fibers are microtubules that come out of the centrioles like this. These little dashed dotted lines. It's almost like fishing line that comes down, or spider web that comes down. If you recall what the centrioles do, what microtubules and microfilaments do is they're like muscles, and they, like we said, they could attach to something and pull it. And that's exactly what's going to happen here, okay? So we have the chromosomes lined up on the equator. These spindle fibers grow down out of the centrioles, and they attach to either side of the replicated chromosomes. So there's a chromatid on this side, and then there's an identical chromatid on this side. Remember, what we're trying to do is make two cells. In order to do that, we have to have two sets of DNA, don't we? And that's exactly what's going to happen. In anaphase, the chromatids are pulled apart. Okay, so we can just show that very easily, like this. So literally those spindle fibers sort of almost reel the chromatids in on either side. So the spindle fibers start to shorten, and because they're attached to the chromatids on either side of the replicated chromosome, they're literally pulled apart. And so now look what's happening. One set of chromatids is going in one direction, and one is going in the other. At this point, when the chromatids are pulled apart, we don't call them chromatids anymore. We actually call them sister chromosomes. Okay. Interesting, huh? And then in telophase, what we have is two new nuclear envelopes forming. Around the two new complements of chromosomes. Okay. Two new nuclear envelopes form. And now look what we've done. We took one diploid replicated nucleus. We pulled the chromatids apart, the identical pieces of each replicated chromosome apart, put them in separate nuclei, right? And now look at this nucleus. Is it haploid or diploid? Well, it's unreplicated, but it's diploid, right? Each chromosome has a partner. There's that one's partner. It's the same size. And there's that one's partner. Same thing here. So we didn't change the ploidy. It's still diploid, but now we have two diploid cells. We started with one diploid replicated cell. We ended up with two diploid unreplicated cells. So the last shot here, the last drawing, is called cytokinesis. You'll notice that this is really nuclear division, right? That's what mitosis is. It divides one nucleus and makes two. Cytokinesis is cytoplasmic division. So really all we have to do cytokinesis is make a wall or a membrane here. 2, 3, 4. 1, 2, 3, 4. And now look what we have. We have two identical cells. Barring any kind of mutation, they are identical in every way, correct? Because these sides are identical due to them being replicas of one another. So we all started out actually as one diploid cell, and then we became two, and then we became 4 and 8 and 16 and 32 and 64. Okay? Or maybe you skint your knee, and this is these are two new skin cells from the mitotic division. Okay, so now where would we be in the cell cycle? Well, we would be back in G1. Both of these cells would be back in G1. Okay, that's mitosis. There's a lot of terminology. There are a lot of C words. You got to, keep watching the video until it sinks in. Read your textbook. It's very helpful. Okay, so now we can look at this image and we can see here's interphase. There's the DNA is still in the form of chromatin. See the thread-like form? And there's prophase where the chromatin has condensed to form the chromosomes. Okay. The nuclear envelope is breaking down. And look, this is just like our cell, two big ones and two little ones, right? Now they're doing pro-metaphase and metaphase, and just forget about pro-metaphase and just go. They're saying it's like early metaphase. Well, that's fine. Here's metaphase. They're all lined up. Anaphase, they're pulled apart. Telophase, and they're showing telophase and cytokinesis happening together. And you can see there's our two new complements of cells. Okay. So I guess the one thing I left out of my drawing really is, and we might as well just put it in so we're clear. And if you want to go back to your drawing, I'm assuming you've been drawing with me. I hope you have. In prophase, we often say somewhere between G2 and prophase, really we would consider early prophase, the chromatin condenses to form chromosomes. So prior to, prophase, you can't really see the chromosomes because it's just in this thread-like form. So it's a minor point, but it's one of the things you could add as something that occurs in that phase, okay? We are doing so well, y'all. We are just making it through. That COVID's not going to get us. Nope. Okay. And here, this is nice because it spells out what's going on in each phase, okay? Chromatin condenses, nuclear envelope disappears, replicated chromosomes line up on the equator of the cell. In metaphase, anaphase, chromatids are pulled apart. Telophase, chromosomes unravel and new nuclear envelopes form. And then here's cytokinesis. Now, keep in mind, if it's an animal, then there's just the membranes, and if it's a plant, then there would be membranes, and then a wall would form in between. Okay. What about meiosis? What in the world is meiosis? Okay. Meiosis is the other kind of reproduction in cells. Meiosis is one part of sexual reproduction in cells. Okay. And so, what does that mean? Well, let's talk about it. Let's understand what meiosis is and what it means. In order to understand meiosis, you have to understand a little bit about birds and the bees, y'all. So let's have a little discussion about the birds and the bees. So here's the thing. If all the cells in your body are diploid, okay, what we need to be able to do, there's two hallmark events of sexual reproduction, and one is called meiosis. Okay, and meiosis, this is sexual reproduction. Okay, meiosis is where you take one diploid cell and you make 4 haploid cells. From one diploid cell, you get 4 haploid cells. We're going to see why it's 4, okay? What does meiosis do? Reduces the number of chromosomes by 1/2, okay? If you got a diploid cell, it basically pulls the homologous chromosomes apart and makes haploid cells. So that's one part of sexual reproduction, meiosis. What are these cells? We call them gametes. And if you're female, it's an egg. The gamete is an egg. And if you're male, it's a sperm. So the products of meiosis are gametes, and they can be sperm or eggs. What do gametes do? By definition, they fuse together to form the first cell of the next generation called the zygote. Okay? Well, maybe this is the egg from your mama. and the sperm from your daddy, 1 plus 1 is 2. This restores the diploid condition in the organism. Every time you go around, okay, and so then there's this one diploid cell, this first cell of the next generation of the organism is called the zygote. We all started as a zygote when our daddy's sperm and our mama's egg fused together. Guess what? From this is one cell, mitosis times many makes the organism, right? Okay, so, and then we're back over here where we started. Where does meiosis occur in us? Testes, ovaries. The only place meiosis occurs in us are in the testes or the ovaries, depending on if you're male or female. Why does this have to happen? Why do we have to do meiosis before we do fusion? Think about it. If you just did fusion without doing meiosis, well, like I said, all the cells are diploid. So you have 2N and 2N. Remember what that means, 2 sets of chromosomes and 2 sets of chromosomes. If you just did fusion, you'd get 4N. You'd get a cell with 4 sets of chromosomes. And then the next generation, you do it again and you get 8N or 8 sets of chromosomes. If you don't do meiosis, then with every subsequent generation, you effectively double the number of sets of chromosomes in the cells, and that's not going to work. No, 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 no. Don't do that. So this is why meiosis is one of the 2 hallmark events of sexual reproduction, the other being fusion. Fusion is easy to understand. It's fertilization in our case. Egg and sperm come together to make the zygote. Meiosis is really what we need to talk about. Okay. So in the next video, we will do meiosis, and we'll then be moving on into the next lesson. Okay. But I think we're going to stop there. I think that's enough info for you. Okay. I hope you have a splendid day. Just so you can see it, there's meiosis. And what I want you to notice about it before we sign off is, look here. Look how these chromosomes have lined up. Now the homologous chromosomes are what are getting pulled apart, not the chromatids in this first part of meiosis. We'll talk a bunch more about it next time. And meiosis is a logical entrance into the next topic, which is genetics. Okay, so we're going to go logically from meiosis into genetics. All right, team. I hope you have a wonderful day. I'm signing off.